But you have enzymes in your saliva called amylase. Most words that end in ASE are enzymes. So you have enzymes in your saliva, and they break down complex carbohydrates. So, you know, crackers are made up of complex carbs, starch. So the amylase in your saliva will begin to break those starch molecules into smaller pieces. Remember that hydrolysis reaction. So it breaks it into smaller pieces, and those smaller pieces, disaccharides, have a sugary flavor, right? So you should notice, so try this sometime, you should notice that that cracker begins to change its sweetness. It'll start to be sweet because you're breaking down that starch and you're releasing sugars. So we're talking about enzymes today, so I thought that would be a good connection if you had one to put on your tongue and experience that, then you could experience that breakdown of molecules. Today we have this our lecture, and at the end of our lecture, because it should be a relatively short lecture, we have vocabulary, but I'm going to have us work on it in teams. So we'll break out into breakout rooms and we can get that done and move on. And then a short exit slip, 5 multiple choice questions. And to finish our time, because flex time is class time, we have an enzyme POGL. It looks kind of big, but mostly I think like the pictures in it are large. And so there's a lot of pictures, which lengthens the whole paper out a little bit. But remember, like Tuesdays and Fridays are 2 class days. So if you were coming Monday and Tuesday to school, we should be able to accomplish this much work. So those days, those assignments do seem a little bit longer, and that's why. But we'll get going on this other stuff, and then we'll have more time for the POGL, okay? Tomorrow, we don't typically have Zoom, right? But because we don't have class Friday, and we have a test Tuesday when you come back, we are going to have a Zoom. It won't be a terribly long Zoom, and it'll be review of material. So we'll have an activity to do. Tomorrow is count day. And so we need to have accurate attendance tomorrow. A lot of times Wednesdays, I'm still checking people like changing their attendance a week later. So I thought if I had a Zoom, I'll get you all in here a little bit better. We can talk about the test and then maybe be more prepared than we were the last test, okay? So I'm gonna, what was that? Oh, let's see, because I normally have that folder with me with the information, but it is not. So I'm gonna go under class resources. And the first thing we have is the daily schedule. So here I'm going to find the answer to our question. Yeah, because the hours are different. Thank you for thinking of that. So this is 4th hour. We meet at 11:06.
Okay, thank you.
That was an excellent question. I'm so glad you asked. I'm so glad you asked. Okay, so the enzyme lecture is still spinning wheels. Do you guys have some background with enzymes already? You talk about them in middle school? Okay, so all the reactions that our bodies undergo require enzymes. So we talked about hydrolysis and dehydration synthesis last week, and those are both going to be on your test. They don't happen without enzymes. Reactions can happen, but it'll take 1,000 years to break down sugar if you waited for it to happen on its own. And we don't have that much time, right? So we need some enzymes to help us get our reactions going more efficiently. So one, we want to build just a working vocabulary to understand metabolic pathways. We're headed into photosynthesis and cellular respiration. They make up metabolic pathways. So we have to talk about the pieces and parts now so we can understand the bigger concepts later. Okay, and then we're going to talk about the enzymes, and you should be able to explain how they work. So when you say explain, you want to kind of, you want to talk about the scientific principles. not just say, oh, they speed things up, but maybe how do they speed things up, okay? So proteins, enzymes are proteins. Remember the last unit we were talking about the organic molecules, and we know proteins are made up of amino acids. So this is what that protein would actually look like structurally. And they function as catalysts, and catalysts speed up reactions. They lower the activation energy. A lot of you identified that in the web quest, or not, the case study with the dog, right? A lot of you identified activation energy, which is the amount of energy that is needed for a reaction to get started. Kind of like if you had, this is like a physical science type of example. If you had a ball and it was sitting at the top of, like, let's say it's a boulder, sitting at the top of the hill. It needs a little bit of activation energy to get it rolling down the hill in the first place, right? Whether that activation is gravity or it's you pushing from behind. There's some energy needed for that follow-up to occur. So that's what activation energy is. So something that is interesting about enzymes is they take part in the chemical reactions, but they are released at the end in an unchanged form. So you still have the same enzymes at the end, and you can use them over and over and over again. Kind of like our money. We exchange hands, and that money can just keep being used over and over again, right? So anything that affects proteins, this is why this is important to know that it's a protein. Anything that affects proteins will also affect your enzymes, right? So enzyme or proteins, they do their job because of their shapes. So if we change their shapes, they can't do their jobs anymore. Anything that ends in ASE, lipase, catalase, sucrase, they're enzymes. The first part usually tells you what they work on. Lipase works on lipids. Sucrase works on sugar, okay? Polymerase polymerizes. So this is showing you the difference in the activation energy, and I think you had a similar graph like this on that gizmo. So if you didn't have any activation energy, it would take, or if you didn't have any enzymes, it would take this much energy in order to get this reaction to start. But with an enzyme, we don't need nearly as much energy, just a little bit, to change these reactants into these products. You should recognize on a graph reactants at the beginning, products at the end. Most of us had those labeled correctly. Activation energy here between the reactants and the products, and then down here, this is the amount of energy that is going to be changed. Energy is stored in bonds. So when you break bonds, you release energy. So the final product here will have less energy than the original molecule, the original reactant. So the whole idea of any catalyst is that it lowers activation energy. And there's lots of different catalysts. Enzymes are biological, meaning made by living organisms. But like heat can be a catalyst. You can be a catalyst when you get like reactions happening between your friends. There's mechanical catalysts, like stirring a solution causes reactions to combine together more quickly than they would if you just let them set, okay? So catalyst in general is just speeding up reactions. And then the activation energy, the minimal needed to cause that reaction to occur. So enzymes will lower that activation energy by holding two molecules in close proximity together. So it's more about positioning. So if this was a synthesis reaction, one that builds, they would hold two molecules close together so that they're orientated properly to have a correct reaction binding. Like if molecules were floating around by themselves, what's the chance that they're going to bump into each other? And then what's the chance that when they do, they're orientated the right way for a bond to occur, right? Thank you. It's a lot easier if you just hold them together and say, here you go, react. Like when you got two friends who are fighting and you're like, here you go, make up, you know, and you hold them together. So there's that active site that is very specific in shape. So like this square, this puzzle piece, they're not going to fit in this site here, right? Only circles will fit there. So we call that the active site. Active sites are on the enzymes. So that's one of the things people can get confused. Where is it? Is it on the substrate? That's what we call these things that bind, substrates. Or is it on the enzyme? So the active site where the action occurs is on the enzyme, okay? These different shapes, the reactants of the reaction, these are the substrates. Complementary means the two fit together, like puzzle pieces. The two will fit together, okay? So the active site has a complementary shape to the substrate. So typically, only one substrate will fit that enzyme. There are some cases where there's more than one that have the exact same shape, and therefore they can both fit there. They can't occupy the same spot, so one might stop the other's reactivity, make it so it can't work. Sometimes they have more than one site, in which case you need both in order for it to work. Okay, so when they bind, we refer to it as the enzyme substrate complex. So up here, the enzyme in bluish and the substrate pink, they'll fit together, and when they do, that's the enzyme substrate complex. That just makes sense, I think. You're either going to break that apart or you're going to put two things together. So here the active site is open. Here's our substrates, the reactants. They will fit in the substrate active site, the active site on the enzyme, making a complex, holding the two in position to cause a reaction. So this would be a synthesis reaction. You're taking two things and putting them together, okay? They can also do the opposite, hydrolysis, break things apart. So in the end, you release the products. So induced fit is kind of an additional concept. Notice the shape here is slightly different than the shape here. So conformational change, conformation refers to shape. Induced fit, once the substrate binds, the enzyme will like hug around it. So either putting two molecules closer together or putting stress on one molecule so that it breaks the bonds and it can break that molecule apart. Okay, so this is showing you catabolism, which is your breaking apart. Remember, think of a catastrophe, it destroys things. So catabolism is a decomposition reaction. Some of you use that word. Hydrolysis is a specific example. Anabolism, some of you use the word synthesis. And dehydration synthesis is a specific example. So here the two are fitting together. And then when the induced fit happens, it squeezes them closer together so they'll bind. Now there's some things that can affect enzymes activity that we want to be familiar with. And we will talk about three. Two of them will change the shape of the protein, the enzyme, and therefore changing whether or not that protein can work. Okay, so temperature, pH, those are the most common, like scientific, like in the lab, they're the most common independent variables we would use to test things, and different chemicals. And I'm not really going to talk about the different chemicals that can affect the shape of the enzyme. But if you change its shape, you can no longer do the function, right? Enzymes, proteins, everything's shape determines the function. So every enzyme has a specific pH and temperature where they work best. I was just talking about the amylase with my crackers at the beginning, right? The pH of my mouth is different than the pH of my stomach. So I'm not going to find salivary amylase in the saliva. I'm not going to find that in my stomach. And my stomach has a different pH than my intestines. So like I'm going to have pepsin, which breaks down proteins, in my stomach, and lipase in my intestines that breaks down lipids, okay? So each of those places have different pHs, so the different enzymes will work there. Different organisms may have different enzymes depending on if they're ectothermic or endothermic, whether they regulate their own body temperature or not, because they're affected by temperature. So both of these will affect the shape. The temperature destroys bonds because it causes molecules to move more quickly. And if you get too hot, they move so quickly that the bonds break. So the shape of the enzyme changes. The pH is a measure of hydrogen. hydrogen is reactive. So the more hydrogen there is, the more likely it's going to bind to the enzyme, causing the enzyme to change its shape. So this is just showing you these different things. So they have an optimal pH they work at. Above or below that, the shape will change due to those bonds breaking. Temperature, same thing, you have an optimal temperature. This is going to increase as you increase because molecules have more kinetic energy. You know, you've learned that before. As molecules heat up, they move more quickly, and as they slow down, or as they cool, they slow down, right? So if they're moving more quickly, they're more likely to connect with an enzyme. So this is going to increase up to a certain point, and then it's too hot, it disrupts the bonds of the protein, and then the enzyme falls apart, denatures. Another thing that can affect is like the enzymes concentration or the substrates concentration, and this should just make sense. The more there are, the more work can be done until a certain point, and then it's once they're saturated, once all of the binding sites are filled, it can't go any faster than that. Okay, so those are 3 things that we could investigate in a lab that would all affect enzymes activities. All right. What questions do you have on that? You can talk about enzymes, the enzyme substrate complex, the binding, the things that can affect the rate of reaction. Good with all of that? Gracias. So I want you to open up the vocabulary that was in that same entry that I was at before. So where we found our lecture, we had some vocabulary. And I'm going to open up that vocabulary. Oh, I didn't talk about cofactors. That's one of them. I did say that more than one. So sometimes like a shape is really big and only the substrate fills half of it. You need a cofactor or coenzyme sometimes that help it fit. So when you come across that word, that's what it is. It helps the substrate fit into the active site. Okay, so open up that vocabulary, and I'm going to put us into some breakout rooms so that you guys can accomplish this as a team. Whether you want to like shout it out, put somebody in charge, or if you want to divide and conquer and then share. It's up to you. Okay, so I'm going to stop sharing in order to form the breakout rooms. Everybody has that open, I'm sure.